Good day, this is Professor Will, CBMD PhD. Today is Sunday, March 1st, 2020. It is 7:35 and 50 seconds a.m. Eastern Standard Time, and this is a short, concise reading on symbiote microorganisms. So let's get started here. Symbiotic microorganisms are the components of the microbiota of an insect that contribute to insect survival, growth, or fecundity. They are born by an estimated 10% of all insect species and are located in the insect gut or tissues, often restricted to specialized cells called bacteriocytes. Most of the microorganisms are rare or unknown, apart from the insect partners, and many have not been cultured in vitro. Historically, the microbiology of insects has been little studied, and until recently, most of the information available on symbiotic microorganisms has been derived from the microscopic analysis of the insect regarding the morphology of the microorganisms, their location in the insect body, and their mode of transmission between insects. The advent of molecular techniques has transformed our understanding of symbiotic microorganisms, allowing the taxonomic identification of microorganisms and the elucidation of molecular bases of their function. Because of insects' dependence on their symbiotic microorganisms, these associations are of great potential value as a novel approach to insect pest management. Symbiotic microorganisms include members of all microbial kingdoms, various eubacteria, methanogens, archaea, and protists, and fungi, eukaryota. The eubacteria, especially members of Y protobacteria, are widely represented both in the guts and cells of insects, but methanogens and protists occur in strictly anaerobic portions of the guts in certain insects. These have been reported in the gut lumen cells and homuncular body cavity of some species. Basidiomyceae fungi are also cultivated in the nest of some insects, e.g. fungus gardening, termites, and leaf-cutting ants, but these ectosymbiosis are not considered here. Most insects have a substantial gut microbiota, although there are a wide difference among insect taxa and among regions of the gut. Much of the literature gives misleading estimates of the microbial diversity in insect guts because the techniques commonly used are based on culturable forms which account for only 0.1 to 10% of the local microbiota. An additional complication in the study of symbiotic microorganisms in insect guts is that many or all members of the microbiota are either transient, i.e. passing through the gut with the unidirectional passage of food, or commensal, i.e. resident for extended periods, but of no discernible advantage to the insect. Detailed experimental study is required to identify which, if any, of the resident gut microbiota are symbiotic microorganisms, i.e. beneficial to the insect. The microbiology of termite guts has been studied extensively. The greatest density of microorganisms is the anoxic proximal portion of the hindgut known as the paunch. In all termites, this region harbors bacteria at 10 to the power of 9 - 10 to the power of 10 cells divided by ml gut volume. All the bacteria are facultative or obligate anaerobes. They comprise methanogens, spirochetes, pilotinas, e.g. Hollandia, pilotina, Dipocalyx, and Clevodyna, and many other identified forms. Another eubacteria including species of Enterobacter, bacterioids, Bacillus, Citrobacter, Streptococcus, and Streptococcus. The lower termites in addition have obligately anaerobic flagellate, protists of the orders Hypermastigia and Trichomonadia, and Oxymonadia at densities up to 10 to the power of 7 cells divided by ml. Approximately 400 species of these protists have been reported in a few including Trichomytopsis, Hermopsidas, Trichomnaphia spirica, both from the termite Zeudermophis, have been brought into azenic culture. Higher termites lack these protists. Apart from the termites, most studies have concerned the gut microbiota of the few insect species that are routinely reared in laboratories. For example, the American cockroach Paraplaneta americana bears both obligately anaerobic bacteria, e.g. Clostridium and Fusobacterium at densities of 10 to the power of 10 cells divided by ml, and facultative anaerobes, e.g. Klebsiella, Yersinia, and bacteroids at 10 to the power of 8 cells divided by milliliter. Locus schizocerica gregaria has exclusively facultative anaerobes, usually at considerably lower densities than the cockroach. And the blood-feeding reduviate bug, Rhodinius prolixus, has diversity of bacteria including Pseudomonas, Streptococcus, Corneobacterium, and various of Actomyces. And not a single gut symbiont, the Actomyces, Nocardia rodnii, is claimed in much of the early literature. The composition of microorganisms in the guts of insects can vary widely with environmental circumstances. The microbiota may change when insects are transferred to laboratory rearings, and specific differences in microbiota of insects in different laboratories, or under different temperature or dietary regimes have been described. Many symbiotic microorganisms are located in insect cells, where they may be well protected from the hemolymph-based defense system of the insect. In any single insect, the cells bearing intercellular symbiotic microorganisms are usually of a single morphological form and location in the insect body. They are called bacteriocytes or mycetocytes, and their sole function appears to be the housing of the microorganism. Most of the microorganisms are eubacteria, but because they have not been brought into a xenic culture, taxonomic information is available only for those forms whose 16S rRNA has been sequenced. Many of the bacteria are members of the Y protobacteria, and the microorganisms in 3 taxonomically disparate insect groups, aphids, tsetse flies, and ants, are particularly closely related. Other intercellular bacteria and bacteriocytes include members of the B protobacteria and mealybugs, and flavobacteria and cockroaches. The bacteria in several insect groups, including the Anoptera and Melophaga, the sucking and chewing lice respectively, and various Heteroptera, have not been studied at the molecular level. Symbiotic microorganisms are widely believed to contribute to the nutrition of insects. This was first deduced from the distribution of the associations among insects. In general, the microorganisms are restricted to insects living on nutritionally poor or unbalanced diets. Bacteriocyte, an insect cell harboring symbiotic microorganism. Nitrogen recycling, the metabolism of insect nitrogenous waste products, e.g. uric acid and ammonia, by symbiotic microorganisms to nitrogenous compounds, e.g. essential amino acids, that are transferred to and used by the insect. Symbiosis, the intimate association between phylogenetically different organisms, often restricted to relationships from which all the organisms derive benefit. Transovarial transmission, the transfer of symbiotic microorganisms to unfertilized eggs in the ovaries of the female insect. Vertical transmission, the transmission of microorganisms from a parent insect to its offspring. One last note, insect hindguts are physically unstable environments. The gut lumen is dominated by the unidirectional bulk flow of ingested food, and many microbial cells pass directly through the gut. Good day, this is Professor Will, CBMD, PhD, and this concludes my short reading. Hope you enjoyed listening. Have a great day.